Hello everyone, my name is Snehlata and I'm a PhD student and a PMRF fellow at the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today, we will continue with the third lecture of the unit, Cell, the Unit of Life. So, before moving to the third lecture, let's first recap what we have studied in our last class. So, if you remember, in the last class, we saw how does a prokaryotic cell look like. So, the prokaryotic cells have three layers, right? It have a three layer that forms the outer boundary, and this three layer together call the cell envelope. So these together call cell envelope. Apart from this, it also has cytoplasm, we studied. Then it has a genetic material, inclusion bodies, organelles such as ribosomes, and certain structure that are present over the surface of the bacteria. We saw a structure something like this. Then we saw another structure something like this. Then we saw these three structures. Now, if you remember, we studied in detail the components of cell envelope. So there were three components of cell envelope. The first one was glycocalyx. The second one was cell wall. And the third was cell membrane, right? So we studied in detail about these components. Now, in this lecture, we will study about the remaining cell structure that are present in a prokaryotic cell, such as ribosome, inclusion body, nucleoids, and the surface structures. So now let's start with the surface structures. So a bacteria have three surface structures. So if you say this is a bacterial cell wall or a glycocalyx, so this is cell wall which has three surface structures. A structure something like this. And what does this structure called? It is called flagella. This structure is called flagella. Then there is one more structure. This is something like this. And this structure is called villi. And the third, a structure something like this. And this is called membrane. So these are the three structures, surface structures that are present over a bacterial cell. Now, we can write these surface structures, or the first one is flagella, second one is membrane, and third one is villi. Now, if you see a bacteria in a microscope, and the bacteria has a structure something like this. So this is flagella. So if a bacteria has flagella, this bacteria will be called motile bacteria. This bacteria is called motile bacteria. Now, if a bacteria is something like this, it does not have any surface structure or a long surface structure, then this bacteria will be called non-motile. Okay. So with this we understood that flagella is responsible for the motility of a bacteria. It is responsible for motility of bacteria. Okay, next. Now, we will study in detail about this flagella and what is the structure or detailed structure of the flagella. So, if you see a bacteria, like I just told, so the bacteria will have flagella something like this. Okay, it will have something like this. But if you will cut it or dissect it and see it under the microscope, its structure will look something like this. So it will have a basal body. Okay, it will have a basal body. This is the flagella I am talking about. Then it will have a tube-like structure, something like this. Okay, so a tube-like structure. This is called hook. And then it will have a long, this kind of structure. And this structure is known as filament. Okay, so a flagella have three parts. The basal body. So the flagella have three parts. Okay, the first part is basal body. Basal body. The second is hook, and the third is filament. Now, this filament is the longest part of the flagella. So, filament is the longest part of the flagella. Now, because it has filament, you can say that flagella is filamentous in nature. Flagella is filamentous in nature. Second thing that you can say about flagella, that it is an extension that comes from the cell wall. So this is cell wall. Okay, and this flagella comes out of cell wall. Hence, it is called the extension of cell wall. Okay, and third, it is made up of a special kind of protein. So this flagella is made up of a special type of protein, and this protein is called flagellin. This protein is called flagellin protein. Okay, so the flagella is made up of flagellin protein. Again, whether this flagella is present in all the bacteria? No. The answer is no. We just saw there are some bacteria in which the flagella is present, and there are other in which it is not present. And this flagella can be of different numbers. Okay, so can vary in numbers. The number of flagella can vary in numbers from bacteria to bacteria. Okay, so it vary in number from bacteria to bacteria. For example, if you talk about cyanobacteria, okay, so cyanobacteria do not have flagella. If you talk about cocci or coccus, you remember the spherical shaped bacteria, so they do not have flagella, or flagella is absent in this bacteria. Now there are certain bacteria such as spirillum. So spirillum, remember spirillum looks something like this. It has flagella only at the top and the bottom. So this is spirillum. Spirillum has flagella at top and bottom of its structure. Okay. Now third is, if you talk about E. coli. Okay. If you talk about E. coli, so E. coli will have flagella all over its surface. So E. coli flagella will be all over its surface. Okay. All over surface, right? So based on the type of bacteria, the flagella, where the flagella will be present and in how many numbers this flagella will be present varies. So here we saw that what is flagella, what are different parts of the flagella. Now one more thing is, if you talk about the basal body consists of 4 rings. So it is made up of 4 rings. Okay, and these rings have certain name. Okay, so the rings name are L, P, S, and M. I'll write here. The first ring is L, the second is P, the third is S, and the fourth is M. So if someone asks that which is the innermost ring present in flagellal basal body, so present in basal body, so it will be M ring. Okay, so the M ring is present in the innermost layer of the basal body. So this is one of the surface structure that we studied. Now the second structure that we'll study is fimbriae. Okay, so before moving, the fimbriae and pili, because we just saw that flagella is responsible for motility. I'll just write here the flagella. It is responsible for motility. But this structure, this fimbriae and pili, have no role in motility. So they have other function, but they do not play any role in motility. Now, what is fimbriae? So if you see, you have a bacteria, something like this. The bacteria have a certain outgrowth, some like this. So this is this hair-like projection. It is called fimbriae. Okay, it is called fimbriae. Now, how does it look? It look like hair-like or bristle-like. So its structure is bristle-like. And what is its function? Its function is it helps in the attachment. So suppose this bacteria wants to attach over a certain surface. So it helps in the attachment of bacteria on any surface. Okay. So this is the role of fimbriae. So if we talk about fimbriae, so fimbriae has this role. Now the first one was fimbriae. The second one is pili. So pili, again, this has no role in motility. So why does a pili need a work? A need? So a pili is responsible or is pili helps in mating. Okay. So in bacteria, as we will study in further classes, the bacteria, there is only one type of sexual reproduction or mating, that is called conjugation. Okay. So you can say that the pili helps in conjugation, and conjugation is a type of sexual reproduction in bacteria. It is a type of sexual reproduction in bacteria. Okay. So pili is responsible for sexual reproduction. Now what it is made up of? So the pili is made up of a special protein that is called pili. Okay. So it is made up of special protein that is called pili. And if we talk about the structure of a pili, so how does a pili look like? It looks like tube-like. Remember, in last slide I drew, so the pili looks something like this. Okay. So it is a tube-like, hence it has a tubular structure. Now, what does this bacteria transfer to this pili? So a bacteria, suppose you have one bacteria, like this, and bacteria have one extra chromosomal material that we will study in later slides, and this extra chromosomal material is known as plasmid. It is known as plasmid. So if a bacteria has pili, it can transfer this plasmid. This plasmid is called F plasmid. Okay. So it is a plasmid that is called F plasmid. So it transfers this plasmid to the pili. Okay. So this is the function of pili. So we have seen the surface structures, that are flagella, fimbriae, and pili. Now next we will move to another structure that we saw in the figure, that is nucleoid. Okay. So what is nucleoid? So nucleoid is, just I said in the beginning of the class as well, that it has, every cell has a characteristic feature that it consists of genetic material. It consists of a genetic material, right? And this genetic material, what does it do? It stores information. This genetic material stores information. And what is the name of this genetic material? The name is DNA. Okay. Now, the DNA can exist in multiple types, such as, one of the example is like this. So it can exist like this. It can also exist like this in this. So this is the linear DNA. This is linear DNA. This is circular DNA. Okay. Now, what does a bacteria have? So a bacteria have a circular DNA. Okay. Bacteria have circular DNA that is called nucleoid. So the genetic material of a bacteria is known as nucleoid. Okay. Now this is where does this nucleoid located? So it is located in cytoplasm. So nucleoid is located in cytoplasm. So it is located in cytoplasm. Now, if you remember, we have studied about the membranes, that there are certain organelles that are membrane bound, then there are certain organelles that are without membrane. So in this case, the nucleoid, whether it has a membrane, no. So it is a naked structure. Okay. Naked structure or naked DNA. So it is a DNA that is not wrapped around, or that do not have a membrane. Okay. Now, whether it is a single DNA or multiple DNA. So when you will study about a eukaryotic cell, you will see that in the eukaryotic cells, there are multiple DNAs. Okay. But in this case, the genetic material is made up of single DNA. So in the case of nucleoid, genetic material is made up of single DNA, or you can also say chromosome. You will study and understand this term in more detail later. But for now, just remember that the genetic material is made up of single DNA or chromosome. Now, this nucleoid is called the main genomic DNA. Okay. So if you remember, I told that the genetic material, what does this genetic material do? It stores the information. What kind of information? So it stores all the information that tells this cell that what kind of chemical reactions has to be done, right? So this it means that this genetic material should have all the information. And in the case of bacteria, the nucleoid is that genetic material. Hence, it is also called the main genomic DNA or main genetic material. Okay. Because why I'm saying main genetic material, you will understand in the next slide. But for now, remember that the nucleoid is called the main genetic material because it have all essential genes. It have all essential genes that is present on this nucleoid that governs that what a cell has to do. Okay. The one more thing that is important to know for now is that the nucleoid do not have histone. So what are histones? So nucleoid do not have histone. So what are histones? Histones are a special kind of proteins, right? So these are the proteins on which the DNA is wrapped. But in the case of the prokaryotic cells, the nucleoids do not have stones. Okay, the nucleoids do not have histone proteins. So it is present in the form of a naked DNA. Okay, so this is a nucleoid that we studied. It is considered as the main genetic material of a prokaryotic cell. Apart from this, we have another genetic material that is present in a prokaryotic cell. Okay, so this was your main genetic material or nucleoid, you can say. This was your main genetic material, and then it also has this a structure something like this that is called plasmid. Okay, now what is plasmid? Again, we have said in the last lecture that nucleoid is a main genetic material. So what are these? These are called extra chromosomal genetic material. Okay, extra chromosomal because these are extra. Okay, yeah, along with the main genetic material, these are the extra genetic material that is present, and they are called extra chromosomal genetic material. Okay, so these are extra chromosomal DNA. Now, what is the shape of this plasmid? These plasmids are also circular in shape. Just like the nucleoid, these are also circular shape and they are also double-stranded. Okay, they are also double-stranded. Apart from this, you can see that the nucleoid is present only in one copy, right? It has only one copy, but is present only once. However, the plasmids can present in multiple copies. So the plasmid can present in multiple copies. Okay. And next thing, unlike the nucleoid DNA that only divides when the cell divides, right? But these plasmids are self-replicating genetic material. So these are self-replicating. Okay. So even when the cell do not divide, this plasmid divide and increases their copy number. Now, what is the role of these plasmids? So if you talk about the role of the plasmids, so the plasmids provide the extra features because as we saw that all the essential genes are present in the genomic DNA, right? Then why do a bacteria need a plasmid? Because it provides the special features. It provides special features to the bacteria. If we talk about what special features I'm talking about, so it is such as giving it antibiotic resistance. Example is antibiotic resistance. Okay, so plasmid is responsible for the antibiotic resistance that is present in the bacteria. So we have studied about the genetic material. One was the main genetic material, that is nucleoid, and the other is extra chromosomal DNA, extra genetic material, that is plasmid. Now next we'll talk about ribosomes. So ribosomes is the only organelle that is present in a bacterial cell, a prokaryotic cell. So this is the only organelle present. Okay. Now, what is the role of this ribosomes? But before moving to this, let me first tell you that how does a protein made up of? Because we know that in our body, everything is made up of protein, right? But how does that protein form? For that, let's see, we have this double helical DNA, right? I just told what is DNA. So we have a DNA And this DNA, or genetic material you can say, it stores all the information that is required by a cell to do its work. But how does this work is translated? For that, the DNA is first converted into mRNA. Okay, so it is converted into mRNA, and this process is known as transcription. So DNA is converted into mRNA, and this process is known as transcription. Now, this mRNA is further converted into the protein. Okay, the proteins that we know. So this mRNA is converted into protein. For that purpose, a special structure, something like this, okay, this structure comes and binds to this mRNA. And what this structure is called? This is called ribosome. And then it starts making a thread-like structure like this, and this is called protein or polyprotein. Okay. So if you talk about what does a ribosome do, we just saw that it forms ribosome. What does ribosome? Ribosome forms protein from mRNA. Okay. So it forms protein from mRNA. Now, where does this ribosome is located? It is located in cytoplasm. Ribosome is located in the cytoplasm, and it is attached to the membrane. Okay, so it is membrane attached. Ribosome is located in the cytoplasm, and it is membrane attached. Next, does it have a membrane bound to it? There is a specific membrane. This membrane I am talking about, cell membrane. Okay, so this is cell membrane. So it is attached to the cell membrane. But does the ribosome has its own membrane? The answer is no. So this organelle is without membrane. This organelle is without membrane. If we talk about the size, so the size of the ribosome is size of ribosome is very small, very small. That is in the range of nanometer. It is of the nanometer range. Okay. Now, you just saw in this structure that a ribosome looks something like this, right? It looks something like this. So it has two subunits. So if you talk about the structure, of a ribosome, then it has two subunits. Okay, it has two subunits. Just like pen, it has a cap and a body. Similarly, it has two subunits. And these two subunits, one is called, it is larger subunit. One is larger subunit, and the other is smaller subunit. Okay. Now, the second thing, you want to measure the weight of this or size of this ribosome. So there is a special unit. Just like if you want to measure something, you have units like kilogram, gram, meter. If you want to measure the length of something, you can measure it in the meter. Similarly, the size of ribosome is measured in a unit, is measured in a special unit. Okay, that is called S. Okay, that is called S. The size of ribosome is measured in a unit called S. So in bacteria, in prokaryotic cell, the ribosome is of 70S subunit. Okay, so in bacteria or prokaryotes, ribosome is of 70S subunit. Okay, sorry, 70S units. Now, if you talk about the larger subunit and smaller subunit, the larger subunit is of 50S. Okay, and the smaller is of 30S. Sorry, 40S and 30S. Sorry, it's 40 and 30S. Okay, so the ribosome is of two subunits, the smaller and the larger. That is, I'm sorry, one second, just one second. So, yeah, sorry. So the larger one is again, so it does not add up, right? So the subunit of the if you see the subunit, it does not add up. So the larger unit is of 50s subunit and the smaller will be of 30s. Okay, so this is what we understood about the ribosome. So it is the organelle that is present in the cell. And what is the function of this ribosome? It forms a protein from mRNA. So all the proteins that are formed by using ribosome. And it is located in the cytoplasm. It do not have any membrane. So it is something like this, which do not have any membrane. And its size is in the nanometer range. And it is measured in a special unit that is called S. Okay. Now, we also come across a term known as polysomes. Okay. Polysomes or polyribosomes. So, as the name suggests, it has two units like poly and ribosomes, right? So, if we talk about how a protein is synthesized in a bacterial cell, so it will have a mRNA. It has direction 5' to 3'. On this mRNA, if you see, there will be multiple ribosomes that will be attached. So there will be multiple ribosomes that will be attached on this RNA, and this structure is called polyribosome. So what is polyribosome? It is multiple ribosomes together form protein from single mRNA. Okay, single mRNA. So there are multiple ribosomes that are attached, and from each ribosome a polyprotein is formed. So this complete structure is known as polyribosome. So here we saw what is polyribosome and why it is called polyribosome, because it has multiple, this poly means multiple ribosomes attached to a single mRNA that together form multiple polyproteins. Okay. Now, next thing that we have to study is about inclusion bodies. So what are inclusion bodies? So if you remember, I said that inside the cytoplasm, it is a bacterial cell. Inside the cytoplasm, there are different small granular structures. Okay. So these granular structures are called inclusion bodies. Okay. And what is the function of this inclusion body? It reserves whatever reserve material a bacteria have. Okay. Whatever reserve material, extra material a bacteria have, it stores it in the form of inclusion body. So you can say that reserve materials or extra materials are present in bacteria in the form of inclusion bodies. Okay. So reserve materials are present in the form of inclusion bodies. Again, this structure is without membrane. It is also naked. So it is without membrane. But unlike ribosome, it moves freely in the cytoplasm. Okay. We saw that the ribosome is attached to the membrane or with the mRNA, but these structures, they move or they stay freely in cytoplasm. So they remain free in cytoplasm. Next, we saw that it is a reserve. It contains a reserve material, hence it is used for storage. So it is used for storage. Okay, now this inclusion body can be of different types, right? Based on what thing it has stored. Based on that, you can name inclusion bodies based on what they have stored. For example, if it has stored glycogen, then it will be called glycogen body. If a bacteria has stored glycogen in it, it will be called glycogen bodies. If it has phosphate in it, it will be called phosphate bodies. And if it has sulfur in it, okay, if it has sulfur in it, it will be called sulfur bodies. Okay. Apart from this, certain bacteria have in their inclusion bodies that is also called gas vacuoles. So gas vacuole is also a type of inclusion body. Okay. And this inclusion body is present in certain bacteria such as in blue-green algae. So the inclusion body such as gas vacuole is present in blue-green algae, in purple sulfur bacteria, in purple sulfur bacteria, and in green sulfur bacteria, and in green sulfur bacteria. So these are the bacteria that consist of gas vacuoles as the inclusion body. Okay. Now, one more important thing, that these gas vacuoles are called pseudo vacuoles. Okay. So these gas vacuoles are called pseudo vacuoles. Why they are called pseudo vacuoles? Because they do not have a membrane. Do not have membrane. And because of this reason, these inclusion bodies, inclusion bodies such as gas vacuole are called pseudo vacuoles. Okay, so we have seen that what is gas inclusion bodies. So inclusion bodies are the one that stores the reserve materials or the extra material that a bacteria have. Now, so till now we have studied about all the structures that we drew in the last class that is present in a typical prokaryotic cell. But now we will see what is the main, based on this now we can see that what is the main difference between the eukaryotic and prokaryotic cell. So there are certain features that I have mentioned, and based on that we can say that whether these features are present in the prokaryotic cell or not. So if we talk about the nucleus, so does a prokaryotic cell have a nucleus? The answer is no. No. But instead of that, it has nucleoid, right? It has nucleus. So what is nucleus? Nucleus is a membrane-bound organelle. Nucleus is a membrane-bound organelle. It is a membrane-bound organelle. But a typical prokaryotic cell do not have nucleus. Okay. Now the second thing, in the eukaryotic cell, yes, in the eukaryotic cell, the nucleus is present. The second structure is the nucleolus. So if you remember the first lecture, I told that nucleolus is a characteristic feature of a eukaryotic cell. So it is present in the eukaryotic cell, but again in the prokaryotic cell, it is not present. So you can say no or absent. Okay, so nucleolus is absent in the prokaryotic cell. Now, if we talk about DNA, so what kind of DNA a prokaryotic cell have? So we just saw in the last lecture, so in last slides, that prokaryotic cell have double-stranded. They have double-stranded circular DNA. Okay, they have double-stranded circular DNA. But in the case of eukaryotic, they have double-stranded. I'm writing double-stranded at the S. Double-stranded linear DNA. Okay, so prokaryotic cell have double-stranded circular DNA, while a eukaryotic cell have double-stranded linear DNA. Now, if you talk about membrane-bound organelles, so does any of the bacterial cell have any membrane-bound organelle? No. So it do not have membrane-bound organelles. So membrane-bound organelles are absent in prokaryotic cells, while in the case of eukaryotic cells, these are present. So we will see in our next lecture that in eukaryotic cells, there are multiple membrane-bound organelles. Now, if we talk about the ribosome, so ribosome is a structure that is common. This structure is common in both prokaryotes and eukaryotes. Both prokaryotes and eukaryotes. Okay, but what is the difference in the ribosome that is present in prokaryote and eukaryote? In prokaryote, the ribosome is 17S. Okay, but in eukaryote, it is 18S. So this is the difference between the ribosome that is present in prokaryote and eukaryote. Now, about the protein synthesis. So where does the protein synthesis take place? So protein synthesis takes place in the cytoplasm. In the case of prokaryote, it takes place in cytoplasm because it does not have any specialized compartment for the protein synthesis. So it takes place in cytoplasm, or you can say on the plasma membrane as well. Okay, cell membrane or plasma membrane. Because you remember, so this ribosome is also attached onto the plasma membrane. So the protein synthesis takes place in both cytoplasm and cell membrane or plasma membrane. However, in the case of eukaryotic, the protein synthesis takes place on the ER, endoplasmic reticulum. Endoplasmic reticulum. Apart from this, if you remember the first lecture, the ribosome is also present in certain organelles in the eukaryotic cells. And this ribosome is responsible for protein synthesis. Hence, the protein synthesis also takes place in that organelles. And those organelles were chloroplast, and mitochondria. So, in eukaryotic cells, the protein synthesis takes place in endoplasmic reticulum, chloroplast, and mitochondria. With this, one question arises that what kind of ribosome is present in the chloroplast and mitochondria. So you will see in the latest lectures that in chloroplast and mitochondria, 70S ribosome is present. Why it is present, we will know later. But in the chloroplast and mitochondria, 70S ribosomes are present. So if someone asks that what ribosomes are present in eukaryotic cell, you will mention both 18S and 70S. Now, next, we'll talk, does this contain thylakoid? So thylakoid is a special structure that is present in the chloroplast that you will see. But in the case of prokaryotes also, this thylakoid is present. Okay, so present but not well defined. Not well defined. We will study in more detail about thylakoids in later. But for now, just remember that thylakoids are present in prokaryotic cells but are not well defined. However, in the case of plants, especially in eukaryotic plants, thylakoids are present, well defined. They are present and well defined. Okay. So they are present and well defined in the case of eukaryotes. Now, if we talk about the envelope system. So what does the envelope mean? It means the outer covering, right? Or a covering. Something like this. So this is envelope. So if we talk about prokaryotic cells, so the prokaryotic cells have only one envelope. That is this cell envelope, right? If you remember, the prokaryotic cell, they have cell envelope that is of 3 layers. So envelope system, it has single envelope system. Prokaryotic cells have single envelope system, while in the case of eukaryotes, it have double envelope system. Why do we call double envelope system? Because in the case of eukaryotes, you have this outer envelope, and then you have cell organelles, right? In the first lecture, I gave you the list of the organelles, and these organelles are membrane bound. So this is the second envelope, okay? So there is one outer envelope, and then there is this second envelope. So in the case of eukaryotes, it has double envelope system. Now, if we talk about true vacuole, so does a prokaryotic cell consist of a true vacuole? You can say yes, but that's not correct, because we saw that, the gas vacuole is present, but this gas vacuoles are pseudo vacuoles, right? So the true vacuole is absent. So prokaryotes consist of pseudo vacuoles, but true vacuoles are absent in prokaryotes. But in the case of eukaryotes, plants do have true vacuoles. So plants are also eukaryotic cells. So in eukaryotes, this true vacuoles is present. Now there is one more term that is called cytoplasmic streaming. So cytoplasmic streaming, what is it? It is defined as movement of cytoplasm. Okay, movement of cytoplasm. It is defined as movement of cytoplasm because of vacuoles. Okay, or around the vacuoles. Around vacuoles. So cytoplasmic streaming will happen where? The one which has vacuoles, right? But the prokaryotic cells do not have vacuoles. So will there be any cytoplasmic streaming? The answer is no. So cytoplasmic streaming is absent in prokaryotic cells, but in eukaryotic cells it is present. Okay. And next, if we talk about the size. So for the size, we have studied in the first lecture itself that the size of prokaryotic cell is very small. Small. What was it? It was typically in the range of 1 to 5 micrometer. But the eukaryotic cells are comparatively larger and has typical size of 10 to 20 micrometer. Okay, so this is a typical size. So these are the differences between the prokaryotic and eukaryotic cells. And with this, you have also revised what we have studied about the prokaryotic cells till now. Now, let's quickly discuss few questions. And now, after that, I'll end this lecture. So the first question is, which of the following bacteria is non-motile? So the non-motile bacteria will be the one that do not have flagella, right? Do not have flagella. So if you remember, I just told in the previous slides that the cyanobacteria is the one that do not have flagella. Apart from this, the E. coli and Spiralium have flagella. So the cyanobacteria are the non-motile bacteria. The second question is, which of the following statement is incorrect about infusion bodies? They are involved in ingestion of food particles. They lie free in cytoplasm. They represent reserve material in cytoplasm, and they are not bound to any membrane. So what will be the correct answer? The correct answer is, what is incorrect about it? That they are involved in the ingestion of food particles, right? Because they are responsible for storing the material. They do not ingest the food particles. The one that ingests the food particles should be present over the membrane. Okay. Now, third is the inclusion body of blue, green, purple, and photosynthetic bacteria are. So we just saw that what are these inclusion bodies called? These are called gas vacuoles, or these are pseudo vacuoles. Okay. So these are the inclusion bodies that are present in blue, green algae, purple, and photosynthetic bacteria. And the next question is that many ribosomes may associate on single mRNA to form multiple copies of polypeptides simultaneously. Such strings of ribosomes are called. So you remember, so you have an mRNA, and you have multiple ribosomes attached to it. Okay. What is this structure called? This structure is called polysome or polyribosome. Okay. So with this, we have completed in detail what is a prokaryotic cell and what are the different structures that is present in the prokaryotic cell. Now in the next lecture, we'll study about the eukaryotic cell. That's all. Thank you.